Hi, everyone. Welcome to the Cerebellar Seminar Series. Today, we have as our guest Robi Soetedjo, who has been absolutely instrumental in describing some of the findings that describe climbing fiber encoding of visual and motor events and their input to the cerebellum. So Robi received his MD from Brawijaya University in Indonesia. I'm sorry, I probably screwed up the name. Then he came to the United States, where he studied with Albert Fuchs and Chris Kaneko at University of Washington, receiving a degree, PhD in bioengineering. With Albert and Chris, he studied the superior colliculus and the role of feedback control during control of eye movements in rhesus monkeys. And with Yoshiko Kojima, he's been instrumental in describing the role of the cerebellum in control of eye movements. Robi is currently a research associate professor in neurobiology and biophysics at the University of Washington. All yours, Robi.
Thank you for the generous introduction, Reza. Our talk today is not about the cerebellum. So I'm going back to my younger self, which is studying the superior colliculus and its contribution to the amplitude increase adaptation. Why doesn't it? Okay. So let's start by reviewing the study by Optiken and Robinson in 1980s. In this study, they performed thanectomy on one eye, here as shown as the weak eye here. And this weak eye is patched after the surgery. And now the normal eye is the viewing eye. So what happened to the saccades? So in response to the 10-degree target step, here shown as a black step here, and the red is the eye, the normal eye reached the target in with a single saccades. But the weak eye may be only about a third of that target amplitude. So now what happened if we switch the patching to the normal eye, and now the viewing eye is the weak eye. So on day one, as you would expect, that in response to 10-degree target step, the weak eye will be short, very short, but it's keep on making a corrective saccade and eventually reach close to the 10-degree target. And the normal eye, as you would expect to, the first saccade will reach the 10-degree target, but the staircase saccade bring the eye position far into the periphery. And then the monkey is returned to the cage and continue viewing with the weak eye. And on day 5, the weak eye actually reached the 10-degree target in a single saccade, but normal eye produce a saccade with more than twice the amplitude of the target. So as you can see from this data that the drive to the weak muscle increases, which bring the weak eye to the target. But at the same time, because the eye is conjugate, the normal eye is becoming hypermetric. So now let's review how saccade is generated. So let's start with the superior colliculus here. The superior colliculus, especially the intermediate and deep layer, they send a projection to the burst generator in the pons and the mesencephalon. But at the same time, it's also branched to the nucleus reticularis tegmenti pontis, which supply an input to the oculomotor vernis, which eventually inhibit the caudal vestigial nucleus. and the caudal fastigial nucleus influence the burst generator activity. And then the burst generator neuron, the burst neuron there project to the motor neuron, which eventually innervates the muscle and move the eye. Many studies, including our lab, have shown that the involvement of this oculomotor vermis and also CFN in the adaptation of saccades. And all of those adaptation was performed in a single day, basically in a single session of experiment. And the possibility that the superior colliculus also involved in the adaptation was also studied, but the result is rather uncertain, with 3 labs, I believe, showing no change in the SC activity during adaptation, and 1 lab showing small changes during the adaptation. So now let's summarize what I'm going to talk in this after this. So basically, I'm focusing on the amplitude increase adaptation, and this is I feel that this amplitude increase adaptation is more relevant to clinically than the amplitude decrease adaptation, because most injuries will produce hypometria. So these are the 4 key questions that I will be asking during this talk. So the first one is that, what are the limits of amplitude increase adaptation? And I think this is clinically relevant. So if we see a patient with, say, for example, that the gain of, the amplitude of the saccade is very small, that it require probably a 4-time increase in the amplitude, and then we ask the question whether that's achievable by the system. So we will try to test how far the amplitude increase adaptation can actually increase the size of the saccade. So now, of course, we will try to understand the time course of this adaptation. And as we have seen previously in this Optican and Robinson experiment, that those monkey was actually performing in a few days. So that would translate into tens or hundreds of thousands of saccades. So, and then of course we will see how the kinematics of the saccade change, the velocity, the duration, etc. And certainly the last one, we will see whether brain region beyond the cerebellum involves. It's probably the superior colliculus. And you will see why I'm thinking that. So next, let's see how we perform the adaptation. So instead of performing tenectomy, which is impractical, and it's probably very difficult to get approval from IACUC, etc. nowadays. So we use a behavioral technique. So basically, the red here is the target that the monkey sees on the screen, a spot of a red dot. And the primary target start to step, and then after some reaction time, so the black here is the eye, the monkey make the saccades to the target. But as soon as the eye start to move, the target jump further. So at the end of the saccade, then the monkey would realize that he is actually undershooting. Then after some intersaccadic interval, he makes a corrective saccades to the new target location. So the primary target amplitude that we will use is between 2 to 4 degrees. And this primary target amplitude here is actually the actual target amplitude, which is not the target step amplitude itself. This is measured from the position of the target at a new location, the primary target at new location, to where the position of the eye at that time. And the reason for performing this is because the step is small, and monkey's fixation is not perfect. So That's the reason for that. So, and then the intra-saccadic step that we use is adjusted online during the experiment, and we estimated from the saccade amplitude, and we aim about 50 to 100% of the saccade amplitude. And between adaptation session, so basically in each session, the monkey will make about 2,000 to 3,000 saccades, depending on the day. And then the animal will return to its cage, and it will wear an opaque goggles to reduce or minimize the visual input, visual feedback that can undo the adaptation. So what's happening? So here, because we use a primary target amplitude of not a single amplitude, so we measure the amplitude of the saccade in a normalized way. So basically we call it a gain, which is simply saccade amplitude here divided by the primary target amplitude. So this is 2 days of adaptation. It starts close to 1, and then if the gain increase close to 2. So now next, after day 8, the gain of the saccade is actually about 2. So now, as you can see, as more and more days progress, so the distribution of the gain is becoming wider and wider, and they are more skewed. So you see more and more saccade with higher gain. What's interesting is that as the days went by, and we can only do this in a that's why we have to stop at day 47 because of just the protocol. So we start seeing that there are 2 kinds of saccades because the distribution of the gains becomes bimodal, as you can see here. So in the first kind of saccades, it saturates the gain at about 2 or 2.5. And we call that a low gain saccades, low gain adapted saccades. And the other kind of saccades, which reach a gain of average about 6 or actually is more than an individual saccade can be as high as gain of 9. So we call this as a high gain adapted saccades. So, and this is the data combination from all 2 to 4 degree targets amplitude. And as you can see here, the trial number to reach this is close to 150,000 saccades. So now let's concentrate on those adapted saccades elicited by 2 degree primary target amplitude. On the left, you will see in the top is a gain, as you have seen as in previous slides. And the second one is the duration of the saccades. The third is the first peak velocity. Here, because why I choose the first peak velocity, because the velocity profile of the saccades as adaptation progress can become a bimodal. So then only the first peak velocity is measured. And the last one is the intersaccadic interval, which means the distance between, or the interval between the end of the primary saccade and the beginning of the corrective saccade. So as you can...
Robi, may I ask a question?
Sure.
Just a clarification. Since the target jumps at most 100% more than the primary target, if the gain is more than 2, wouldn't that be also another error? So I don't understand how come they go up to gains of greater.
Yeah, good. That's a good question. I've actually, this is the first time I'm performing this experiment. I realized that actually when they did the corrective saccade, of course, that's kind of, you undo the adaptation from the previous saccade, right? The primary saccade, because that intrasaccadic step is large. But nevertheless, they will still perform this adaptation. So, and in we refine this technique. So in the new technique, it's actually after the intrasaccadic step. So as soon as we detected the corrective saccade, we blank the target. Basically, we blank the target for 500 milliseconds, and that seemed to speed up the adaptation a little bit. So yes, but even with the way it is now, it is actually still adapting. And eventually, what happened is that I think what happened is that as your saccade get larger, there are a big dissociation between the primary target step, which is small, which is 4 degrees or less, and the intrasaccadic target step is becoming so large. So you can imagine, right? Let's take 2 degrees, but when the gain has to reach 7, so the intrasaccadic step is actually 12 degrees. So it's really large. So that's becoming outside of the adaptation field that I will talk later. So okay, so where am I? So I was talking about the intersaccadic interval of between the end of the primary and the beginning of the secondary saccade. So as you can see, one of the thing that is strange here is that the bimodality of the gain is actually also shown in the bimodality of the saccade duration. So this suggests that the duration plays a big role here to make that high gain saccade. That's one. And then the reason that why the low gain saccade saturates is actually because the first peak velocity saturates. And then one unexpected that I see this is that actually the intersaccadic interval decreases over time. And then eventually some corrective saccade appears in a much shorter latency, which in this case about 100 milliseconds, and some appear even shorter in less than 50 milliseconds. This gives us a clue what's actually happening that give us an idea, well, is that actually the saccade actually merging? So let's see on the right column. So this shows first 50 of day 1. So we see that this is, we can consider that as the pre-adaptation saccade. So you can see that the velocity of the saccade is slightly above 100 degrees per second. And you also see the corrective saccades here in the velocity profile. But now at day 3, in the last 100 saccade, you see there's a big increase in the peak velocity. And of course, that peak velocity increase is followed by duration increase too. And the pink traces here is actually always from the previous plot as we go on this figure. So as you can see that there is not much change in the latency of the corrective saccades, but there's probably a few that is actually earlier than the one in the day 1. So now let's go to day 9, last 100. So the peak velocity continually increasing. Saccade is still a single distribution. And now, but you can start seeing that you have at least a couple of these have a very short latency corrective saccades. And most of the corrective saccades also closer to the primary saccade a little bit. But as you go to day 21, you start seeing a small population of the high gain saccades, and also large population of the low gain saccades. As you can see here in the velocity profile, peak velocity doesn't change a bit. So, and on the green traces there is two examples of primary saccade velocity that fall almost to a stop, but after that, it reaccelerates again. And the reacceleration is actually happened earlier. And then as you can see that, again, you have big population of short latency corrective saccades and longer latency corrective saccade. Now, as we go on day 31 and then day 47, as you can see, we see less of the low gain saccades and more of the high gain saccades. And again, as you see, reacceleration after initial movement is becoming more prominent. And the three green traces also shows you, again, reacceleration that happened just when the velocity was actually decelerating to a very low. As you can see also some corrective saccade with latency of less than 50 milliseconds. And then as on day 47, you see that most of the data is actually on high gain saccade and with very little population of low gain saccades. And then as you can see, then also the saccade reaccelerate in most of them and still showing some of the short latency corrective saccades. And again, velocity, peak velocity that doesn't change very much. And this is for 2-degree primary target amplitude. And the same thing, the same kind of pattern you will see also for 3-degree target amplitude and 4-degree target amplitude. So now we'll see the adaptation transfer. So as you recall that we adapted 2 until 4-degree saccades, which is shown in blue here. So when we tested the 1-degree saccades, we see a lot of transfer. So the gain is still high. And for 5-degree saccade, the gain is still high, and you still see population of high gain saccades, the same as gain of 6. But as the gain reaches, oh, I'm sorry, I should talk about target amplitude. For 5-degree target amplitude, you see still a lot of high gain saccades, the same for 6-degree target amplitude. By the time you reach 7-degree target amplitude, most of the saccades are low gain, and the gain is still high at 8, 9, and then it's essentially gain written to 1 for 10-degree saccades or more. And then, of course, the adaptation also generalized or transfer to other direction. We adapted at 0-degree, which is rightward horizontal, and it's transferred well to 15-degree, plus minus 15-degree of horizontal direction. And at 30-degree is mostly low gain saccades, and the gain is still more than 1 at plus minus 45-degree. So now we'll test, as we have done in the past with Yoshikos and Albert, we test how does that, we adapted basically visually guided saccades. So we test how does it transfer to, I think, express saccade or gap saccades or to delayed saccades. So in this express saccades, so when the monkey is fixating, the target is off. And then for about 150, you have to adjust that 150 to 175 millisecond. And then the target appear in the periphery and on the amplitude that we wanted. And then the monkey make the saccades. This paradigm will create a lot of saccade with latency of 90 millisecond or less. And that's what we shown here in the red. But in the delayed saccade, the way we generate the saccade is that while the monkey is fixating, the target appear. So at the same time, while the fixation is still on. And it is continually on, so both target in the new location and the fixation for 600 milliseconds. And then after that, the fixation light goes off. And that is a go signal for the monkey to make the saccades. So as you can see here, so the visually guided saccade here is already adapted. So in the long-term amplitude increase adaptation. So the target here is actually 2 degrees. So as you can see, the amplitude of the express saccade is much larger. It's actually transferred so well to express saccades. But really, essentially, during delayed saccade, the amplitude of the delayed saccade is like the low gain saccades, which is has a gain about 2, which is here. That's about between 0 to 4 degrees is about gain about 2 or a little bit more. So now let's look at the velocity profiles. As you expect that, as I've shown you previously, that the adaptation will produce a reacceleration for the high gain saccade. And that too, reacceleration is becoming more prominent and actually started earlier during express saccades. But that's reacceleration actually absence. It's not there during delayed saccades. So see here the dichotomy of the transfer. That's meant to express saccade. That's what make us think then, well, perhaps the superior colliculus might have something to do with this creation of this high gain saccade. So let's review what we learned. So after about 30 days of adaptation, saccade gain distribution become bimodal into low gain and high gain population. And low gain saccade result from the saturation of the peak velocity. High gain saccades emerge through the merging of the primary and the corrective saccade. This is what I thought, because as evidence from the shortening of intersaccadic interval. So in a way, we can say that high gain saccade is initiated by a low gain movement and followed by reacceleration that increase duration and of course amplitude. High gain saccade transfer to the express task, but not to the delayed task. So our hypothesis here, the superior colliculus contribute to this process through a division of job. The rostral side initiate the low gain movement, and therefore the neuron should burst for both low and high gain saccades. For the caudal superior colliculus, they provide additional drive for reacceleration during high gain saccade. So we expect that the activity is much higher for high gain saccade and maybe no or very little activity for low gain saccade. So let's review the colliculus quickly from Robinson 1972, the stimulation study. And the study showed that colliculus has a topographical map, that the rostral is encoding smaller saccade. This is the left colliculus. And the more caudal you go, you have larger saccade. And the more medial is upward, and the more lateral is downward. And that actually can be fitted roughly by this map here. And this is an example of a single unit recording from a visual motor neuron. After 2 degree target step, this is unadapted. So you see a visual response about 70 milliseconds later. And then when a big burst happen, and then when you have the saccades. And then you can plot the number of spike against the size of the saccade vertical and horizontal component of the saccades. As you can see, this number of spike peaks about 2 degrees and then slowly roll off for larger amplitude, but no activity for ipsi movement. And actually the roll off very steep in the inside edge. That's mean from the very small to the big. So now let's see the rostral visual motor neurons. So here, because as you know that the saccade has been adapted, right, during this test. So we have to test the side of the colliculus, not using the eye movement, because it's already distorted by the adaptation. So instead, we use a visual response probe. It's simply the monkey is fixating, and then you flash the target at different location. So that's what we see here. This neuron actually responds best maybe around 1.5 degree of the, this is the visual response of the neuron for during the probe. And no saccade is produced during this time. And of course, as you expect, and then there's really no response at all at 4 degree and beyond. I just didn't plot it. And so then if you present a big target like say 8 and 10 degree, as you expect, no activity. It doesn't respond. But now we presented the target that we use during the adaptation, which is within the peak of the visual response of the neuron. So this is actually the reason why we decided to choose a small target amplitude so that we can have a big dissociation between the movement and the target amplitude. So we align on the target step. As you expect, we see visual response in both low gain and high gain adapted saccade. And then we align on the saccade onset. During low gain, you have a big burst. Well, this is, even though the saccade is actually rather large, it's at least about close to 4 degree or so. And then during high gain saccade, which has the amplitude probably about 10 degree, it discharged almost similarly. So here the dash magenta here is from the low gain activity. As you can see here is actually during high gain adapted saccade, the activity of the neuron is slightly lower, but it is also longer than during the low gain saccade. As it's probably expected that because the saccade has a longer duration. So now we show that we confirm our first hypothesis that the rostral collicular neuron bursts for both low and high gain adapted saccade. Now what happened with the population of the neuron? So basically what happens, saccadic bursts increase its duration with amplitude. So this is all the saccade elicited by 3 degree or less amplitude, because that is the tuning of the preferred amplitude of the neurons. So the data here is sorted on amplitude of the saccades. The top here has a on average about 2.1 degree amplitude, and the gain is about 1.5. And at the low end here, the amplitude is about 20 degree, and the gain is about 7.8. As you can see, and this is aligned on the saccade onset, the peak of the activity is normalized for each neuron, so that we see here that the peak of the burst is higher for low gain saccade, but as amplitude becoming larger and larger, the peak burst is becoming somewhat lower. But at the same time, you can also see that the duration of the burst is keep on increasing, and this is confirmed by the end of the saccade. So the green dot here represent when the saccades end. As you can expect, as amplitude get larger, the duration of the saccade get longer. And that's basically confirmed that the increase in the duration of the burst is related to the increase in the saccade duration. So now let's go to the caudal neuron. In the caudal neuron, as you expect, this neuron is during visual response probe, it's about 10 degree at the peak. So here we presented at 8 degree target step. It shows big saccadic motor burst. And you can also appreciate some of the visual response here. But now what happened during low gain and high gain adapted saccade. During low gain adapted saccade, there is very little activity. But that activity actually increase during high gain. So now you would probably expect that it will be gradual, and it is. This is an example for the 3 neuron that we have. So the data again is sorted on saccade amplitude. Here the black, and the brown here is actually plotting the primary target amplitude. They are all 2 degrees or less. So we wanted that because we don't want to contaminate the motor burst of the colliculus, the saccadic burst of the colliculus, with a possible visual response. So, and then of course, as you expect, because it's sorted on saccade amplitude, the gain here, which is on the left column, is also increasing. And as you can see here from the raster, that for this is for the same target, that when the saccade was small, it's basically almost no activity or very low activity. The third neuron has some irregular resting rate. So basically there is no burst. But as amplitude get larger and larger, or gain get larger and larger, you see that there are more and more activity. There are more and more number of spikes. So we can then summarize it as a population. So we pull the rostral and the caudal neuron. The rostral neuron is shown here in the dash. So we selected trial that have a gain that give us mean gain about 6.7 and mean gain of 2.8 to represent the low gain and saccade here. And this is for the high gain saccade, and this is the velocity profile. As you can see that actually the first 20 milliseconds until about the peak, the velocity profile is actually indistinguishable. And this is the activity of the neuron. So for low gain saccades, there's a big burst with high peak for rostral neurons. But for caudal neuron with a solid curve here, there's very little activity. But during the high gain saccade in the magenta here, you have reacceleration, and the rostral neuron also bursts maybe about 40%, no, it's about 60%, 50-60% of the peak activity. But at the same time, the duration is also longer. Actually, I did look at the number of spikes within the saccades, and they're actually a constant for the rostral neurons, or some of them actually increasing a little bit. But for caudal neurons, you start seeing more activity, much higher than during low gain saccades. And the peak is about later than the peak of the rostral neuron. But you can see that the activity is small. It's maybe about 60 spike or 70 spike per second. Is it significant? Actually, is it actually causing the reacceleration? So to test that, we perform an inactivation study. So we use an injectrode here that we designed, and using the tip here has a 35 gauge stainless steel tubing, and it is inserted into a thin wall 30 gauge tubing, stainless steel tubing, and they are all insulated, just like we make a recording electrode basically. And then we expose this part to make a contact with an amplifier or electrical stimulation, and then we continue with the Teflon tubing, and where we injected 2% lidocaine. And we aim on the larger side on the colliculus, 10 degree or more. So remember that we have to rely on the visual probe to identify the site of the injection. So here is this injectrode allow us to record multi-unit activity. And actually, sometimes we got lucky that we can isolate a single perhaps is a big neuron in the colliculus. So we have a visual response here, as you can see. And then online, we have to perform this online, and we just run a trigger level. And then we get, we plot it against the target amplitude. And we see that this neuron is actually like 10-degree appearance of the target. And then after that, we confirm it with the stimulation. As we know, because 10-degree, the adaptation does not transfer to 10-degree saccades, 10-degree target amplitude step. So we perform electrical stimulation. And consistent with the recording, the electrical stimulation produces 10-degree eye movement, 10-degree saccades. And then we injected the lidocaine, and the saccade to the 10-degree target post-injection is actually much is smaller and slower. Well, it's smaller because it is slower. So now what happened with the adapted saccade? So because the injection is located at this 10-degree, we try to, and as you expect, the drug will spread, right? So we limited the saccade to dose elicited by less than 2-degree amplitude. And during this recording and injection, we actually make the adaptation to include 1 degree, so 1 to 4 degree actually that we adapted. So this is the gain of the 2-degree amplitude. And we let it run about 15 minutes, about 1,000 seconds. And then we perform the injection, the downward arrow here. And as you can see, the high gain saccade start to disappear and then disappear. And then, because this is lidocaine, it is recovering there. So now what does it look like for the saccades? So this is before the injection. This is where the red here is. And this is the velocity profile of this saccade in the red dash here. As you can see, there's a lot of reacceleration and also a lot of corrective saccade. And then now, what happened when the site is inactivated? We see almost complete disappearance of the reacceleration. And you don't see these corrective saccades anymore. It's likely becoming a much longer latency. And then, as the drug dissipated, we see reappearance of the reacceleration. And then we average these traces. And as you can see, the drug doesn't affect the peak velocity, the first peak velocity. So that means the initial movement is not affected, but it's only affecting the reacceleration of the saccade that actually produced the high gain saccade. So then in conclusion, so that inactivation supported our hypothesis. So then we conclude that the high gain saccades require caudal SC activity, and high gain saccade reflects a combined mechanism, basically. The rostral SC initiate the low gain movement, and the caudal SC provide an extended displacement command through reacceleration. And pharmacological inactivation of caudal SC selectively impairs the high gain saccades. And the take-home message is that the superior colliculus doesn't just initiate saccade, it can actually involve in the plasticity through recruitment of the more caudal sites to extend the movement command and to increase the amplitudes. So I have on time, so it's 8:46 now. I have more slides that I can show, but it's kind of hard to place in this talk because, but if you don't mind, I can show it to you a little bit.
Let's go to questions. Let's see if we
Okay.
have some questions, Robi. Maybe in the
Okay, sure.
context of questions, you can
Yeah,
You
sure
can.
that.
Yeah, so please just go ahead and raise your hand or however you like. We can ask questions from Robi. So let me begin, Robi, and ask you about that interesting observation that you had, generalization to express saccades, but not the delayed overlap saccades. What do you think is partly, I guess, what I'm thinking is that what do you think is the role of the cerebellum? Sorry, the colliculus in that generalization that you saw and that lack of generalization for the overlap, the delayed saccades?
Well, the colliculus is certainly firing for during delayed saccade, right? I mean, so, but it seemed to me, looking at all this data, that all of this happen because, say, this intersaccadic interval gets shorter. So anything that has a shortening has something to do with the initiation of the saccade itself, and it's that it has that in a short latency. So that suggests that the colliculus is actually might involve this kind of the thought. And certainly maybe that during the delayed saccade that more planning, because you have to suppress the saccade, right? I mean, during delayed saccade, you have to suppress your urgency to make the saccade. So it's probably it relies more. I'm not saying that express saccade doesn't rely on the cortical input, but maybe during delayed saccade, you have more reliance to the cortical inputs to actually make the saccades happen. You have to release your suppression. You have to, and that might actually might change the way the how the colliculus is activated. That's just I mean, a wild guess on my part.
Right. All
So,
right.
well, and the other thing also that's this is not actually surprising too, in a way, that memory guided saccade is also, and in some part, a delayed saccade too. And this actually we tested on memory guided saccade is actually the same thing that doesn't transfer, except that the data, as you expect, memory guided saccade is so variable. So memory guided saccade actually doesn't really transfer to I think that's correct, yes, that to visually guided saccades and express saccade for that matter. And I think delayed saccade also has a minimal transfer to express saccade. So I guess that's supporting the idea. Although we can never know for sure about that. It's probably a very difficult experiment to know that. That's the best I can answer that.
That sounds good, Robi. David, you have your hand up.
Yeah, thank you very much. Well, for a clinician, this was spectacular. And I mean, scientifically too. But so I have just a few comments. Number one, the analogy for me in the clinic has to do with how we use saccades to adapt to a low gain VOR response. And as you all probably know, that when you move your head and if your vestibulocular reflex gain is not right, the slow phase is not matching head velocity, we use saccades to get on target. And if you look at the type of adaptation that takes place, it's so similar to what you're describing because this is, again, like you said at the beginning, it's low gain is the problem. And you see over days how the patient will gradually incorporate saccades that are not driven directly by the visual error at the end of the head movement. They're called covert saccades.
Yeah.
And the latencies change, the distributions change. And it's very analogous to me in some respects to what you're doing. And of course, we're always wondering, where do these corrections come from? How can we promote them in vestibular patients? We used, you may know, we used the later LATER analysis that Roger Carpenter has put forth as a nice way to show how the distributions change. And we always kind of thought that maybe it's somehow related to express saccades because the latencies get less and less. So that's really quite interesting. Two other quick points. The reacceleration I thought is fascinating because it reminds us if you do a double step in a patient who makes slow saccades, they start out and then they slow down and they actually can pick up when the brain suddenly is being apprised of a change in where the target is. Maybe this is just all coming from the cerebellum and driving the superior colliculus, and that your colliculus lesion is not really proving the adaptation takes place there, but it's a conduit of the adaptation downstream. One last point. The model of using muscle weakness versus the model of using double target steps is different in one important respect, and that's what might be the role of proprioception. Because you have a proprioceptive efferents-Capri mismatch when you have a muscle weakness. And yet when you use the double target step, which is kind of a little bit imperfect for muscle adaptation, there's no mismatch between proprioception and efferents-Capri. So the adaptive mechanism, if indeed proprioception plays a role, might come in more to the muscle model than the double step model. So anyway, that's all I have to say today. Just a few small comments. It's really interesting stuff.
Thank you, David. Yeah, so actually I've thought about that because how the colliculus knows that it has to, to trigger reacceleration. And actually, part of this is actually kind of I'm jumping too far from a baby step. I was thinking already that maybe if I performing some optogenetic experiment, that, say for example, putting a channel rhodopsin in the CFN and then perturbing the colliculus through the projection basically placing the optical fiber in the colliculus and see what happens. So some kind of a future experiment that I have in mind. Yeah, so I think it might be that the colliculus might receive a signal from the cerebellum. So we don't know yet.
Great, thank you.
Yeah.
Robi, may I ask you a question?
Yes.
Yeah, first, beautiful work, really, really interesting data. So in our marmoset monkeys, in a very small portion of saccades, we see these double saccades that look like your double steps, but obviously we don't have any adaptation task there. And we see them we have the random corrective saccade task, we see them both for the primary, for the corrective saccades and the back to center saccades. So for all the amplitudes, there are these portions that when you look at the velocity profile is double peak, as if something is stopping the eye then moving on. So I think you have a very nice paradigm to show that colliculus is involved in this, if it is the downstream of cerebellum or not. But I want to ask what you think about do you see this in macaques too? And what do you think if this is happening in real time as well?
Yeah, we see, I mean, when you blank the target, basically you turn off the target. So when the saccade, when the monkey is making saccade to the target, actually many times the monkeys knows that they are actually, say, hypometric. So they make a corrective saccade. And many times the corrective saccades also have almost a continuous of movement. And that's actually, as David also mentioned, that during a double step paradigm, you see that too. So in some trial has basically zero intersaccadic interval. Yeah, we see that. We see that in macaques that, from time to time that the monkey just, the saccade is just like have a dip in velocity and then reaccelerate again. It's just this paradigm make it actually happen consistently.
I see. So we see that in mossy fibers that we believe are from colliculus because they have movement fields, we see double peaks too,
Yes.
which is kind of similar to what you have with like longer durations in the activity. But I was wondering if you looked at your data, how actually colliculus responds, because even if it is that, if what we get in the mossy fibers are from colliculus, they're indirectly from the NRTP. So I'm wondering if colliculus has enough information about those double peak saccades to categorize that this is like solely is being driven from the colliculus rather than like cerebellum directly have an effect on the double step, or yeah.
Yeah. So probably I have to show you this slide. I mean, actually what happened during the merging, right? Is that basically a fairly describe your question? What is the activity of the colliculus as the primary and the secondary saccade get closer and closer and merge, right? So, okay, let's go there. So here we have, this is a kind of hard analysis to do because we have actually another monkey that actually makes a lot of less than 50 millisecond intersaccadic interval. But this is what we have basically. So here is a recording from a caudal SC neuron. You would expect that during this time that the rostral colliculus is active, right? It's bursting because of the small target step. So this neuron is probably tuned about maybe 9 degrees or so. So here we try to match on all these three conditions as much as we can do to do this analysis. So we see here there's very little activity for the primary saccades, right? That you expect this is a caudal neuron and the target step. And that actually I will mention, I will show you later. I think that's important data that I show. Anyway, so then you see this little activity as you have seen earlier. And then the monkey will make a corrective saccade with a longer latency, let's say 100 milliseconds or more. Actually, the one that has longer latency, you see here some visual response too. That is for these two saccades here. And you see this, it bursts just like normal, you presenting maybe that's roughly about 6 degrees. So like you presenting 6 degree step. And this is how the neuron would fire for that saccade, right? But now you can see as this is kind of unexpected from the colliculus for me. But normally I would expect that the colliculus activity would be always congruent with the saccades, right? At least the motor burst, I mean, what I mean activity here is the saccadic burst. It will always be consistent with the saccades. So here, a similar corrective saccade happened as in this. It might be slow, a little bit slower, but the latency is much shorter, it's 50 milliseconds or less. As you can see, the dot here, I'm sorry, I have to tell you, the dot here, well, it's probably shown here, is represent the onset and end of the second corrective saccades. So it's way lower than that, basically. It's basically changed the behavior. It's basically becoming more similar to the activity during primary saccades. I mean, initially you would expect that because the saccade will bring the eye to the intended goal, basically to the, and you should expect this to fire similarly like that, right? But it is not. And then as you merge more, so you're becoming one, and that activity is becoming basically overall activity. I cannot distinguish where the beginning of the corrective saccade. So they're becoming more activity here. And this is, I think, why the caudal SC neuron actually activity increases during the making of the high-gain saccade when the two saccades are merged. Does it answer your question?
Yeah, this is very interesting. Thank you so much.
Are there
So
any other,
it's
are
that.
there any other questions for Robi?
Yes.
Florian, please go ahead.
Hi, Robi. Great work, really. Super interesting. I have a question regarding the other eye. So if I understand your paradigm correctly, the other eye is occluded,
No.
or
Well,
is kind of the visual input is
it's
attenuated.
both. No, because we use a behavioral task instead of muscle weakening.
Yeah.
So the two eyes actually viewing. It's just we trick them behaviorally. Basically, the monkey is fixating, and then the target jump. As soon as the eyes start to move, the
Yeah.
target jump further.
Yeah.
So the two eyes are viewing.
Okay, the two eyes are viewing, but if I understood you correctly, the non-weakened eye basically over time overshoots.
Oh, I see. You're talking about this. So let me go back.
Yeah, very early on in your talk.
Yes. Okay.
So the non-weakened eye kind of overshoots the target, right? As the weakened eye learns to do the
Yes.
corrective saccade.
Yes.
Have you observed over your adaptation period in the weakened eye, have you observed any interocular adaptation so that the overshooting eye over time reduces its overshoot error?
Well, I cannot answer your question because I didn't use this thing because I didn't use thanectomy on our monkeys. We just use behavioral technique.
Mm.
Yes, definitely. When you have both eyes, is that your question is when you have both eye viewing, correct? So I guess then you will have, depending on whether or which eye is actually more dominant. I mean, in some patients, they actually, I think one study from John Hopkins too, I forgot, that actually the dominant eye is adapting and they're just ignoring the hypermetric.
I can comment on that if you want.
Oh yeah, you can probably comment.
Yeah, so we did a little disconjugate adaptation in both monkeys and humans by using selective prisms in one eye versus the other eye. And yes, there is some disconjugate adaptation as well. The conjugate adaptation seems to be more powerful,
Yeah,
but certainly if there's a range where the brain can kind of fuse these disparate images, you can show some adaptation in the saccadic system.
okay, thank you.
Wonderful. Robi, that's really an
So-
incredible experiment that you've done.
Yeah, so this is the last one that I want to show you. I guess probably that's actually related to Amir's question, I believe. So yeah, so now you ask the question, right? I mean, we always like to think about the colliculus neuron as have a response field, right? Either we call it movement field or visual field, etc. So here we show three different way of analyzing this. The top one is using visual probe that they're just flashing the target at different location while the monkey is fixating. But in the second row, it's actually we plotted the saccadic burst. It's not the visual, like the first row. The saccadic burst that against the primary target amplitude. And then, as you can see, and we just artificially separated the high gain saccade as saccade with a gain more than 3, which is the pink here, and the green is a low gain saccade with gain less than 3. As you can see, if you plot them against primary target amplitude, which is mean the visual, basically, right? They actually have a tuning very similar to the visual response probe. Well-defined tuning. But if you plot them against the saccade amplitude, again, because the adaptation distorted the saccade so much, and you can see you will have a broadening of the, of the plot. So that's actually indicates that
It's very interesting, Robi. This is actually what I wanted to like follow up on, and
Yes.
that is probably if we had something like calcium imaging, probably
Yeah.
what you might have is that your neurons have more muted response, but more neurons across these amplitudes are being activated
yes.
that are
Yes.
pushing the eye forward to. And kind of, I think your last row is showing that if I'm correct, because the neurons are responding for the saccades that are not related to their movement field, but probably more of them are active with lower rates because it's not their preferred amplitude. But
Yes.
the population is pushing forward the double peak.
Yes, exactly. That you see here. Basically, this is the caudal neuron, right? So for the high gain saccade is slowly increasing as saccade get larger and larger.
Yeah, this is beautiful. Thank you so much.
You're welcome. Bye-bye.
Wonderful. Well, thank you, everyone. Robi, thank you so much.
Thank you.